Welcome! This is Regular Pitch Talks, Session 2. Learn about fascinating research and don't forget to vote for your favorite presentation. First up, please welcome Roxane Drainville from Université de Montréal.
Incomplete spinal cord injuries are associated with chronic motor deficits. The recovery of locomotion generation and control over the affected legs are possible and can account for great improvement in quality of life. Many studies demonstrated that spinal and brain stimulation approaches can treat motor disorders related to spinal cord injuries. However, none has compared their single and combined effect to see if it has a greater immediate modulation of walking. This is why our lab developed a new neuroprosthesis that stimulates the brain and spinal motor circuits in synchrony with walking, which means that we deliver the stimulation at the precise movement of the leg. To do so, we implanted 6 rats with electrodes on their hind limb muscles, in their hind limb motor cortex, and on 2 spinal segments, lumbar and sacral. After this procedure, we collected baseline data of rats walking in real time on a treadmill, and we then performed an incomplete spinal cord injury to obtain paralysis and foot drop on their left leg only. The plan was to evaluate the immediate effects of cortical and/or spinal stimulation over treadmill locomotion in the intact state and after spinal cord injury. While the cortex is central to voluntary movement control, the spinal cord generates patterns of muscle activity to generate walking. So how does our cortical spinal neuroprosthesis works? Because the spinal cord injury is incomplete, there is still a possible connection between the spinal cord and the motor cortex, which can be recovered and improved by our neuroprosthesis. Also, its design allows to stimulate at the best time to have a maximum effect on the leg with the deficit, and has shown greater results in modulating movements than continuous stimulation. The electrical activity of the muscles in the intact leg was used to predict the gait cycle of the deficit leg. This makes it possible to stimulate at a precise moment with the characterization of stimulation parameters such as amplitude, frequency, and duration. Both cortical and spinal stimulation, when delivered in synchrony with the left foot lift, increased the leg flexion, and when it was delivered 80 milliseconds after the foot lift, it increased leg extension. Moreover, the increase of amplitude stimulation at these specific timings allows the neuroprosthesis to have a maximum effect in step height and to reduce dragging before and after spinal cord injury. These experiments demonstrated that cortical spinal neuroprosthesis has the potential to reduce motor deficits and enable rehabilitation protocols after spinal cord injury.
Thank you, Roxane, for your presentation. Next up, we have Justine Fortin-Houd from Université de Montréal.
Hi everyone, in our project we are interested in the hippocampus, well known for its role in memory formation. Sharp wave ripple are high amplitude, high frequency events representing the synchronous activity of hippocampal neurons. Sharp wave ripple are present during slow wave sleep and immobility period and are really important for memory consolidation. Many brain regions project to the hippocampus and can modulate sharp wave ripple activity. One of these regions is the median traphe, which is well known to be composed of serotonergic neurons. But the median traphe also contains glutamatergic neurons, expressing the vesicular glutamate transporter type 3. These neurons, glutamatergic neurons, project to the hippocampus. Part of our study was to characterize this glutamatergic pathway from the median traphe to the hippocampus. To investigate the role of these VGLUT3 neurons, we use VGLUT3 cream mice and Cre-dependent adeno-associated virus to express our optogenetics tool specifically in VGLUT3 neuron. So we inject a virus expressing channel rhodopsin, so an activated opsin, in the median traphe. So our glutamatergic neuron express this opsin. We implant an optic fiber over the median traphe to bring the blue light to these neurons. Simultaneously, we record in the hippocampus with electrodes so we can record the sharp wave ripple while activating the glutamatergic neuron in the median traffic. You have an example of a short slow wave sleep recording shown here, and you can see our detection of the sharp wave ripple event. Below, it's our main result. So in this graph, at time 0, it's the time of the stimulation. Here, it's the stimulation of 24 pulses of light of a duration of 10 milliseconds at 20 hertz. And you can see that the sharp wave ripple frequency drops to 0 when we activate the glutamatergic neurons from the median traffic, meaning that the activation of these neurons suppress sharp wave ripple activity in the hippocampus. We did shorter stimulation protocol and saw the same result. Also, you have a controlled experiment where we inject a virus that doesn't express the opsin, and we do the same protocols, and we saw no effect in these control mice. So these results suggest an important contribution of this median traffic hippocampal glutamatergic pathway on memory consolidation that we look forward to investigate.
Thank you for sharing your work with us, Justine. Next up, we have Refilwe Mpai from McGill University.
Child is father of the man. I understand this quote by William Wordsworth to mean that lessons learned during childhood, both positive and negative, guide us into adulthood. They mold and shape who we become in later life. This phenomenon is especially true for the brain. Experiences that occur during critical periods of plasticity shape the trajectory of brain development. A critical period is a period during which brain circuits involved in a particular function are especially receptive to receiving information. In some cases, instruction from the environment is absolutely necessary in order for normal brain development to occur. At the end of a critical period, plasticity in the brain declines significantly and learning is much harder. This is mediated by perineuronal nets. So, what are perineuronal nets, or PNNs as I will henceforth refer to them as? They are a mesh-like structure of condensed extracellular matrix made of various proteins and sugars that surround cell bodies and proximal dendrites. The proportion of neurons that are encapsulated by a perineuronal net increases as the brain reaches maturity. These PNNs reduce the ability for new experiences to leave a permanent trace in the brain. It is believed that adverse experiences that occur during these critical periods of plasticity, such as child abuse, result in deviances from typical neurodevelopment. This increases one's predisposition towards mental illness later on in life. In fact, people with a history of child abuse are twice as likely to develop major depressive disorder and 3 times more likely to develop anxiety disorders. So with all this in mind, we hypothesize that child abuse disrupts connectivity through the altered recruitment of perineuronal nets in brain regions involved in emotion regulation in people who suffer major depressive disorder. So in recent years, the cerebellum, which has largely been considered a motor region, it has been implicated in emotion regulation and cognition, as well as major depressive disorder. So through my project, I hope to explore neuroplasticity through perineuronal nets in the context of child abuse and major depressive disorder in the human cerebellum. I am currently characterizing perineuronal nets in postmortem human cerebellar samples in healthy controls using immunofluorescence. After that, I will then compare PNN expression between cases and controls. So with this project, we hope to bring insight into the mechanisms of vulnerability and responsiveness through the lens of early life adversity on brain development. My name is Refilwe Mpai, and thank you for listening.
Thank you, Refilwe, for your talk. Next up, we have Alexandre Thibodeau from Université Laval.
Imagine a typical morning. You wake up, dress up, drink your coffee, leave for work, take your car, have an accident, never walk again. Imagine peripheral nerve injuries are a serious health concern and leave most patients with lifelong disabilities. Fortunately, axons have the ability to regrow after an injury, but to do so, we have to give them a little help. Nerve tubes can be used in clinics to repair peripheral nerve. In fact, nerve tubes act as a bridge to connect both sides. The axons have a physical support to grow from the proximal segments of the injured nerve to the distal segments. In our lab, we think that the living nerve tubes made solely from patients' own cells will promote a better healing. In the laboratory, we can seed endothelial cells that will form in vitro a capillary network and be able to rapidly connect with patient vasculature. So, quickly after graft, the nerve tubes will be efficiently oxygenated and increase the cell's survival. With a small patient biopsy, smaller than a $2 coin, we can extract fibroblasts and endothelial cells. We can multiply the cells and seed fibroblasts that will produce their own extracellular matrix and self-assemble into a manipulable tissue sheet. We seed on it endothelial cells that will self-arrange into a capillary network and adjuvant cells that will promote axonal migrations. The sheet is then rolled up to form a tubular structure ready to graft. To evaluate the potential for motor function return, we graft our tubes to repair 15 mm sciatic nerve injuries in a rat model. We did a 22 weeks follow-up. We observed three main themes: the vascularizations, the axon migrations, and motor function recovery. First, the tube is rapidly vascularized after graft with blood flow inside the pre-seed human capillaries. So we can tell the vascular network created in vitro had connect to the rat network after graft. Also, there was no sign of anoxia in the center of the nerve tubes. Second, we looked at the nerve fiber migrations inside the tubes. Most of the axons were able to completely cross the tubes and recolonize the distal segment of the rat. So the nerve tubes did its jobs to support axonal migrations over long distance. Third, we look at the motor functions. Rats must walk on a horizontal ladder with rugged and variable distances to quantify the locomotor movement skills. The rats crossed the course and we scored their walk. The results showed that the nerve tubes had a significant return of motor functions in a rat model. It has been shown in a long gap rat model that a living prevascularized and pre-seed Schwann cell nerve tubes could allow a better motor function recovery. These nerve tubes could provide a new clinical tool to repair much larger lesions.
Thank you for your presentation, Alexandre. To conclude the session, we have Zoe Ward from McGill University.
This isn't exactly what it looks like. Contrary to popular belief, Viagra does not only have one target audience with one target issue. In simple terms, Viagra increases blood flow. And when we think about it that way, a whole world of therapeutic opportunities is introduced. Blood is an invaluable carrier of oxygen and nutrients that gives our bodies the energy they need to function. But what happens when blood can't get to where it needs to go? Picture a lush garden. Every plant is thriving, shady undergrowth beneath the trees, vibrant flowers. This garden can be as successful as it is because of its irrigation system, a complex network of plumbing that provides water and minerals to every square root of greenery. One day, though, the water stops. The garden recognizes this lapse and starts taking measures to protect itself. But as these dry conditions continue, the greenery droops, leaves are lost. The undergrowth is the first to go, and with it, the nutrient balance of the garden's ecosystem. Once green, now brown and yellow, and eventually the plants will die. The water can even be turned back on, but sometimes the damage is too great and the garden will never be like it was. Now map that fairly bleak image onto a brain. Specifically, a newborn brain deprived of blood and oxygen at the time of birth. This is called birth asphyxia. The support cells called glia, like the undergrowth, are some of the first to die. Neurons in surrounding areas follow suit. Even when blood is restored to the brain, much damage remains. Birth asphyxia is devastating and not at all uncommon. It affects 5 in 1,000 children in developed nations and 26 in 1,000 in developing nations. Just like the garden, a newborn brain can sense that it is in trouble when there is even the slightest reduction of blood and oxygen. The nervous tissue reacts quickly but can accidentally cause more harm than good. In an effort to slow this unhelpful reaction, total body cooling has been used. Living in Canada, we know just how slowly things will move if they're cold. And it's a similar concept here. While cooling treatments do save lives, Many of the asphyxiated babies still have brain damage and go on to develop neurologic disorders. We need a therapy that can work alongside cooling, one that increases blood flow, restoring that essential irrigation system. Enter Viagra, and the basis of the work from the NeoBrain lab that I'm a part of. Data from our lab has already shown that in baby rats, Viagra used alone has promising restorative effects on neuron regrowth and tissue repair. But the picture is still blurry. I want to know now how Viagra works alongside existing cooling therapies. My theory is that cooling and Viagra will work in complementary ways. Cooling will slow some of the inherent neural pathways that accidentally promote damage, while Viagra will increase blood flow, oxygen, and nutrients to support cell survival. Through my work, I hope to bring us one step closer to that thriving garden, one step closer to securing a better neurologic outcome for babies asphyxiated at birth. Thank you.
And this concludes our regular Pitch Talks session 2. Please don't forget to vote for your favorite presentation.